Here's a simplified, diagrammatic version of part of the DNA chain. It still looks pretty complicated, so let's see how it's built up. There are two main strands, wound round each other in the form of a double helix. Each strand is made up of phosphate groups, here coloured yellow, and deoxyribose groups, coloured green. The two strands are linked together by basic groups which are attached to the deoxyriboses. The bases on one strand are joined to those on the other by hydrogen bonds. 4 different bases are involved: cytosine, which can only link with guanine, and thymine, which can only link with adenine. The bases can only pair in this way: cytosine with guanine and thymine with adenine. You can see that here. C always linked with G, A with T. So, DNA consists of long-chain molecules in which 2 spiraling strands are linked by pairs of bases in the way we've just shown. The bases may occur in different orders in different DNA molecules. Now, an important feature of DNA is that it can replicate itself, make copies of itself. This is how it does it. The 2 strands come apart. Each separate strand now builds up a new strand alongside. The new strand is built up from separate nucleotides, each a combination of a phosphate group and a deoxyribose group to which one of the 4 bases is attached. The base finds its complementary base on the DNA strand. Here the base is thymine, which links up with adenine. Here, cytosine links up with guanine. And so on, until a DNA double helix has been formed once more. The same thing happens to the other strand from the original DNA molecule, so that now we have 2 DNA molecules. This is how a given DNA molecule can make any number of copies of itself. How does DNA carry genetic information so that a particular plant or animal develops in the way it does? Slightly simplified, it works like this. The spiral unzips again, and this time a slightly different strand builds up alongside the DNA strand. This is ribonucleic acid RNA in a form called messenger RNA. It's like DNA, except that the green deoxyribose groups are replaced by ribose groups, here coloured orange, and at the same time the base thymine present in DNA is replaced by uracil, here coloured purple. The sequence of bases along the RNA strand matches the sequence of bases along the DNA. This messenger RNA, and remember this is only a short portion of a very long chain, now moves out from the cell nucleus where the DNA is to the ribosomes in the cell. Here, proteins are built up. This is how the process works. Molecules of transfer RNA, each with groups of 3 of the 4 bases, attach themselves. Each carries a particular amino acid depending on the sequence of the 3 bases. This one with uracil, adenine and cytosine carries an amino acid represented by the grey rectangle. It finds its place by base pairing on the chain of messenger RNA. A chain of amino acids is built up, each amino acid corresponding to a particular triplet of bases or anticodon on the transfer RNA, which fit the corresponding triplet or codon on the messenger RNA. The genetic code is a triplet code controlling which amino acid comes at each point in the chain. The sequence of amino acids in the chain has been determined by the sequence of bases in the original DNA of the cell nucleus. The amino acid chain now peels off from the RNA. It's a polypeptide, part of a much longer molecule, which is a protein. What particular protein it is has been determined by the sequences of bases in the original DNA. A gene is any portion of the DNA chain which affects a significant part of the primary sequence of a particular protein. Genes regulate the synthesis of proteins, and the proteins, structural and enzymatic, determine the characteristics of any living organism. So DNA, which controls protein production, therefore determines the characteristics of the plant or animal in which it's present. Now, any change in the DNA carried by an organism will mean that a gene is changed. As a result, a different protein will be produced on the new code carried by that gene. Suppose, for example, that due to chance exposure to radiation or to some mutagenic chemical agent, a thymine group is lost and replaced by cytosine. This is the messenger RNA chain which the DNA programmed before the change, but it no longer fits because cytosine doesn't link with adenine. The new messenger RNA will have no adenine but guanine in this position. In the cell ribosomes, this altered messenger RNA will now build up a slightly different protein. The transfer RNA with the anticodon guanine, uracil and adenine, carrying an amino acid represented by the pink rectangle, no longer fits at this point, because uracil will not link with guanine. A different unit of transfer RNA with the anticodon guanine, cytosine, adenine does fit, but of course it carries a different amino acid, the green rectangle. The polypeptide now carries a new amino acid at one point. We have in fact a changed primary sequence in the protein. A mutation, a change in the DNA, in the gene, and certain changes cause much more drastic effects than the example we've taken,